Good evening and welcome to the live practice session for the Environmental Biotechnology course, and we are in the week 10. So we will start the session now. Uh, could you please confirm if the audio is clear and the screen is visible? Okay. Could you please confirm if the screen is visible and you can hear my audio? Okay. Assuming that I am audible, I will start the session now. So until the last session, we have seen about the techniques in environmental biotechnology and about bioremediation, biodegradation. So we'll continue our discussion. Um, so the first topic that we'll be seeing today is regarding biodegradation and indigenous microorganism. Uh, Biodegradation is a biologically catalyzed reduction in the complexity of organic compounds where microbes interact with organic chemicals and they result in reduction of their molecular complexity. So what we essentially mean is the organic compounds are going to be complex and those are going to be the pollutants basically. So in our case, we are going to use the microorganisms to degrade these complex organic compounds, and we are trying to reduce the molecular complexity and hence by the toxic effects that is uh, exerted by these organic compounds. And in this process, we have the breakdown of complex structures like double bonds, single bonds, substitution bonds, and all these together result in the oxidation. And as a result of oxidation, there is going to be release of electrons. Uh, the microbes will use these electrons in their own electron transport system for energy generation and other benefits. So basically, the organic pollutant is going to be oxidized, as a result of which there is going to be some electrons, which the microbes will use it for its energy synthesis and growth. Indigenous microorganisms present within contaminated sites or natural environments exploit the biodegradation of various organic compounds as a metabolic niche to obtain energy and building blocks for synthesizing new cellular material. So what this statement means is now we have directly gone into talking about indigenous microorganisms. So something that belongs to a place uh, is called indigenous. So the microorganism is naturally existing in a particular habitat, and hence we call it as a indigenous microorganism. So when an organism is present indigenously in a site, it means that the microorganism has the capability to utilize the organic compound or the organic pollutant, or to tolerate the organic compound or pollutant. So in either cases, the microbe is going to thrive. If the microbe is not tolerant, it is sensitive, then the microbe cannot continue living there. So its survival becomes a question mark. So when we find some indigenous microorganisms in a contaminated site, then we can conclude that mostly the organism is using the contaminant as the energy source, for its survival. And these microorganisms are naturally occurring in that environment, which means that they have the suitable metabolic uh, capability to survive there. So metabolic capability means all the proteins necessary uh, for structural integrity, for functional integrity. So for functional integrity, it's mostly going to be enzymes which can break down the pollutants that are present in that contaminated sites. So it means that the microorganism is completely ready for survival in that contaminated site. So now we'll talk about what are the bacterial metabolic requirements for biodegradation. So bacterial metabolism requires four fundamental things. One is the energy source, one is the carbon source, one is the electron donor, and next is the electron acceptor. So we have energy source, carbon source, electron donor, and electron acceptor. These requirements uh, are basically not exactly same for all the microorganisms. Every organism needs energy source, carbon source, electron donor, electron acceptor, but not the same energy source is there for all microorganisms. So based on the permutation and combination possible with this energy source, carbon source, electron donor, and electron acceptor, we have a high range of metabolic diversity in nature, which leads to microbial diversity as well. When microbes are going to interact with these pollutants, they are going to generate enzymes that can degrade the pollutants and oxidize them, and this process is known as catabolism. So breaking down something is known as catabolism. And in this oxidation process, the electrons are extracted from the organic compounds, and there should be some carrier molecules. So electron donor is going to be our organic pollutant. So until it reaches the acceptor, there should be some carrier molecules that carry the electrons from this donor to acceptor. So mostly we have this NAD plus, NADPH, and FADH2 as the uh, electron carriers in microbial systems. So we have already seen about catabolism, where it is about breaking down compounds. So mostly oxidation takes place, where the organism secretes some enzymes which can break down the organic pollutant or oxidize the organic pollutant, and from here it gets the electrons. So and next comes the anabolism. Anabolism is basically uh, enzymatic biosynthesis. So biologically something is getting synthesized or built up. In catabolism it is about breakdown, in anabolism it is about building up. So anabolism uh, is where these microorganisms are going to perform biosynthesis using various enzymes. And one more key difference between catabolism and anabolism is catabolism is a breakdown and hence it releases energy, whereas in anabolism you have to build something, so it is energy intensive and it is an energy consuming process. So catabolism is a breakdown process, and when you break down, energy is released. In anabolism you're going to build something for which you have to spend energy. So these are the key differences between the concepts of catabolism and anabolism. While talking about metabolism, metabolism is the combination of catabolism and anabolism, where microbes deploy degradative enzymes for catabolic reactions, while they also facilitate the anabolic reactions through biosynthetic enzymes. So the other requirements include micronutrients like phosphorus, nitrogen, magnesium, etc. Okay. So until this catabolism anabolism part we have discussed in the last few minutes of our last lecture. So from here on we will go with some new topics for this week. So first of all, the factors affecting the rate and extent of biodegradation. The rate and extent of biodegradation depends on several critical factors that create significant variability in the process. So the primary factors that affect the rate and extent of biodegradation. So rate, in the sense we are talking about how speed or how fast the reaction is happening, the biodegradation process. That is the rate of the process. And when we talk about the extent, it is about how efficient the process is. If there is 100% of pollutant that is present initially, how much are we able to degrade? Whether we have 90% efficiency in degradation or 50% efficiency in degradation, that is the extent of biodegradation. So in any biodegradation process, you will take into account both of these: how fast the degradation is happening and to what efficiency is the degradation happening. These are the two key things that you have to consider. And one of the factors that influence this rate and extent of biodegradation is the microbial community composition and catabolic abilities. So when can a microorganism degrade something faster? Whenever it has the metabolic capability to do that, right? The microbe is not eating slowly, like that is not the context in which we would talk scientifically in environmental biotechnology. It is because how fast the enzymes can act and how plenty the enzymes are, all these are the factors that determine the rate of the reaction. So the type of the microorganism present and their catabolic abilities will control the rate and extent of biodegradation. So we have other factors also, like environmental factors. So the prevailing environmental condition present the most challenging factors, including the availability of terminal electron acceptor and other essential nutrients like nitrogen, phosphorus, and trace elements required by microorganisms. Temperature, pH, and redox condition of the environment are critical for the biodegradation process. So the astonishing diversity of microbial capabilities and variability of site-specific conditions make it difficult to predict the degradation rates and extent easily. So in environmental factors, we have seen about the availability of electron acceptors, other nutrients and physical factors like temperature, and also factors like pH and redox reaction is going to affect the rate and extent of biodegradation. We also have a limitation based on the um, site where this degradation is going to happen. So that is the site specific variability. So as we saw in one of the primary factors, which is the microbial community composition, the diversity of organism is often unknown and it is also variable. First thing is it is unknown, and when we try to characterize, it is not going to be the same everywhere. So the diversity of organism is unknown and local factors control both the biodegradation process and microbial community composition. So this variability invalidates the extrapolation of experience from one site to another. So that is what I was telling. Yeah, if you try to characterize one spot for, okay, this conditions are present, these microorganisms are present, a spot A is going to be characterized, but you cannot extrapolate the same thing to spot B also. Uh, so that is where this site specific variability comes into picture. Just a minute, I have some fluctuations with my internet. I'll just fix this. Okay, so let's continue with this. Um, the factors affecting the rate of extent of biodegradation. So the next one is the need for environmental interventions. While many microorganisms are interested in degrading organic pollutants, um, they also have other requirements beyond carbon and electron source. Microorganisms need other nutrients like nitrogen, phosphorus, magnesium, iron, sodium, potassium, calcium, and many other macro and micro elements along with the carbon source and electron donors and acceptors, because these um, are going to maintain the transport uh, intermembrane transport, and other homeostatic processes are going to be controlled by uh, the environmental factors. So interventions involves providing the supporting factors when they are not naturally available or optimal. This So when we are going to have a biodegradation process, we have to check if that process is suitable for the microorganism to first survive, and then only it can go for degrading. So if the survival itself is a question mark, then there is no point of degradation there. So if a particular site is not good for this degradation, then the microbe cannot survive there. So the intervention involves providing the support factors when they are not naturally available, or when they are available at a very low concentration. So we can supply terminal electron acceptors like nitrate or iron. We can supply macro elements and micro elements which serve as these acceptors also. And one more thing that we can do is we can go about adjusting the environmental conditions so that the microbes can survive in that environment. Like we can adjust the pH, we can make it acidic, we can make it alkaline based on how the microbe can survive, or we can go about controlling the temperature. So this is where we shift from a natural degradation process to a process where we go for a reactor kind of setup, where you have all this fixed, all the pH fixed, temperature fixed, and you have a very monitored process. Sometimes the natural degradation proceeds efficiently without human intervention, but when the conditions are suboptimal or they are below optimal, intervention is very essential to support the microbial growth and stimulate the degradation process. So now we will see about the two-phase model of biodegradation. So the two-phase model of biodegradation represents a fundamental concept on how the microorganisms can process organic pollutants. So the biodegradation process can occur through the following sequential reactions, and these two reactions are not overlapped. Like, yeah, they are connected, but they are distinct. That is the meaning of two sequential but distinct metabolic phases. So in phase A, you have specific degradation where the pollutant specific enzymatic transformation happens. So when you have a antibiotic, an antibiotic specific degradation happens. When you have some pesticide or you have a dye, a very enzymatically specific degradation process occurs. That is phase A. So at the end of phase A, you will have some common products which can go into a central metabolic system. So imagine you have a core in between, and from wherever you start, you reach the core, and inside that core, you have another reactions for energy synthesis and stuff. So that is how this two-phase system works. In phase B, you have the central metabolic system, where at the end of phase A, you will have a link to connect phase A and phase B. So the product of phase A will be the starting point of the central metabolism, and that converges to a common point. And this division allows microbes to handle diverse pollutant structures while channeling all degradation products through common metabolic networks. So as we saw in phase A, we have pollutant specific enzymatic transformation. So we have unique enzymatic pathways for every pollutant, and it's based on the structure of the pollutant molecule. And when we have major structural modifications, the pathway is going to keep on changing. And the main aim of phase A is to convert this process or link this process to a central metabolic intermediates. And the funneling process that channels, it acts as a funneling process that channels the diverse compounds to a common point. So the key features here are, first is the substrate specificity. Different pollutants require different phase A pathways. Like aromatic compounds follow distinct initial step. Aliphatic compounds with varying carbon chain use different enzymes. Polyaromatic hydrocarbons PAH require multi-step breakdown process. So the phase A is very substrate specific, and you have a high degree of enzyme complexity. So just in case, like we saw about this poly aromatic hydrocarbons, they require a multi-step process. So in a multi-step process, you require different enzymes in each of the step, which is a multi-step enzymatic process. Specific enzyme induction is required for every pollutant, and you have sequential intermediate formation through dedicated biochemical pathways. So the end product of the phase A is going to have the connection to the phase B. So the major products that you can expect most commonly are pyruvate, acetate, and acetyl CoA. So pyruvate is the major endpoint for aromatic compounds. Acetate, you can get it from different degradation pathways, and acetyl CoA is where through which it's going to enter the central metabolism. And here are few examples that you can go through for phase A. In case of phase B, which is the central metabolism, you are going to integrate into universal metabolic pathways. So the primary pathways here include TCA cycle and other fermentation pathways. So complete oxidation of acetyl CoA and pyruvate happens in the TCA cycle with energy generation. In fermentation, occurs generally in anaerobic process where oxygen is limited, and it produces products like organic acids, alcohols, and gases. So next is the integration and energy flow between phase A and phase B. So first we have this metabolic integration where the transition from phase A to phase B occurs through product recognition, which means that phase A products are recognized by phase B enzymes, and there is a seamless integration in this process. There is no lag, there is no gaps that has to be addressed, and there exists a high quality energy coupling. Phase B provides the energy for phase A. So phase B is essentially going to have reactions in which you will have energy generated, and from that energy phase A is again going to circle. So this is the energy coupling between phase A and phase B of biodegradation. And coming to the energy and material flow, you have again a seamless flow of carbon where the pollutant itself is going to act as the primary carbon source, which converts into the central intermediates, and these are going to then be assimilated as the microorganism itself, and hence you have this biomass plus carbon dioxide. And in case of complete mineralization, the pollutant is going to be degraded into carbon dioxide and water. And talking about the energy flow, you have ATP generation from phase B. So as we saw, we have TCA cycle and electron transport chain in phase B. which are energy generation processes, and this can again be spent in phase A, where there is energy requirement to break down the pollutant molecules. So here we also need to talk about the cellular organization and regulation. Um, the process can be uh, orchestrated by a group of microorganisms, or it can happen only by the action of a single group of organism. So when it is all about a single organism degradation, we just have one microbial strain, not only bacterial but any microorganism for that matter. We have one bacterial strain that performs both the phases, and we have complete degradation capabilities in individual cells. And next we have the syntrophic degradation, where the division of labor between different organisms exists. We have a group of organisms, and there is an assigned task for each group of organism, and they have a metabolic relationship where the product generated by one organism can be utilized by the other in a series of steps. And we can also find interesting factors like interspecies metabolite transfer, like hydrogen acetate can be transferred from one organism to another organism. This is also part of the syntrophy. And then you have enhanced degradation through cooperative metabolism. You have methanogenic partnership utilizing fermentation products. So the fundamental prerequisites for microbial degradation of organic pollutants uh, is a very much studied topic. So the understanding of the fact that biodegradation begins with recognizing the process as a self-purification process is very important. So when we talk about a self-purification process, it refers to the natural process in which the microorganisms are there in the polluted or contaminated site. They slowly get acclimatized to that environment, to the concentration of contaminants, to the type of contaminants, and they slowly start degrading the contaminants as well. So any microorganism for that matter, like bacteria, archaea, fungi, all of this can adapt to the pollutant and can naturally be present in a contaminated site. So like the examples given here, contaminated soil, polluted groundwater, wastewater treatment plant, all these processes are going to be self-purified. So this is what our first assumption was. There is going to be any microorganism that can cause the biodegradation of any pollutant. So keeping that in mind, any contaminated site will have its own indigenous microorganism that brings about the self-purification process. And the role of indigenous organism here is to get naturally acclimatized to the contaminants, to gradually adapt and grow in the presence of that pollutant, yeah, and then to develop a specific degradation pathway and capability over a time. It's kind of evolution of the microorganism to survive in the polluted environment. And finally, once the individual organism is ready, it has to interact with other microorganisms to form a self-assembling groups with relevant biocatalytic activity. So that is where you find this syntropy coming into picture. So for this, you need the presence of specific degrading organisms with appropriate metabolic capabilities. So a site contaminated with pesticides should have microorganisms that can degrade the pesticides and not any other dye or anything. You can have diversity in its function, but their ability to degrade the uh, pesticide should certainly be there. So, when we talk about reading the genetic capabilities, as we saw in one of the previous lectures, when we analyze the DNA, you can potentially tell this gene is present, so the protein or the enzymes can be produced, and hence this microorganism can perform a certain type of biodegradation. But when we talked about the significance of transcriptomics, we were clear that we need to study which genes are expressed, which are functional. So the functional enzyme expression and production is the key to biodegradation. It does not matter whether the organism is having the gene or not, but it should be really metabolically capable of synthesizing the enzyme and then carrying out the biodegradation process. And there should be proper conducive environmental conditions for the microbial growth. So even though the microorganism is capable of degrading organism, the temperature is going to be so high that the microbes can tolerate, or the pH is going to be too acidic, then the microbe cannot survive in that environment. So it's a challenge to the survival of the microorganism itself. Hence, we need a conducive environment in which the microbial growth can, and there should be adequate contact between pollutants and microorganisms. So the microbial degradation process can occur through natural attenuation or engineered bioremediation. In natural attenuation, we are just going to have indigenous microorganisms, and we are going to allow them to degrade uh, the pollutants. Whereas in engineered systems, we go for biostimulation or bioaugmentation. So in the case of biostimulation, we will be adding some nutrients to stimulate the microorganism and to um, fasten the process. In bioaugmentation, we are going to add some more specialized microorganisms that can aid in the faster degradation. The success of biodegradation depends on the presence of capable microorganisms and their ability to express their functional enzymes, which are having the potential to break down the pollutants into simpler non toxic forms. So, next we will be talking about the multi-enzymatic nature of biodegradation process. So, until now, if you have any doubts or suggestions, you can post in the chat box. I'll wait for a couple of minutes. Okay, so now we'll discuss about the multi-enzymatic nature of biodegradation process. So biodegradation is a very complex process, and it's mostly a multi-enzymatic process. It's not like you just have a reaction where A gives B and yeah, your complete degradation is over. No, it's biodegradation doesn't happen that way. It is a multi-enzymatic sequential process where you have one step after the other, each having its own energy gain or energy expenditure, and each have being catalyzed by a specialized enzyme. So this kind of complex reactions are essential for the complete breakdown of organic pollutants into uh, simpler compounds. So the multi-step nature of biodegradation uh, typically involves multiple enzymatic steps. As it is mentioned here, you get B from A and then C from B, and you have some 10-15 steps to get the final product. So one example is the benzene degradation pathway, where the benzene or benzoate is taken up by the microbial cell and it is initially oxidized. And then you get go about benzoate conversion to phenol and phenol to catechol, and then you have the conversion from catechol to semialdehydes, and these semialdehydes are further um converted to oxopentanoic acid derivatives, and then these oxopentanoic acid derivatives are going to be converted to acetaldehyde and pyruvic acid. So when you talk about the entire process, it is going to be benzene conversion to acetaldehyde or pyruvic acid, but it's not direct. You go about multiple steps to achieve this conversion of benzene to acetaldehyde or pyruvic acid. So we talked about phase A and phase B in biodegradation. So in phase A, you have the conversion of pollutant to central metabolites. In phase B, the central metabolites are then integrated into the central metabolic pathways to give complete oxidation along with generation of some energy. So when we talk about this multi-enzymatic process, each step involves specific enzymes. And you can classify these enzymes using EC code or the enzyme classification numbers, and you can find it commonly in many databases. Another important aspect in biodegradation is the bioavailability. So bioavailability is a critical factor that limits biodegradation efficiency, and bioavailability essentially is the accessibility of the pollutant to microorganisms. So we have a pollutant, a contaminated site, and we have microorganisms. It doesn't mean that the degradation is going to happen directly. So you have to check um, whether the microorganism is capable of accessing the target pollutant, and only then it can go about oxidizing it or breaking it down. So this accessibility of the pollutant to microbe is known as the bioavailability. So bioavailability is the degree to which the pollutant molecules are accessible to the microorganisms in the aqueous phase, and how effectively the microbes are able to interact with this pollutants. So microorganisms quite often assimilate these pollutants from the liquid phase, and then only they go with degradation, not degradation directly. So let's see some of the factors that control bioavailability. One is the physical sequestration. So the pollutants that are trapped in micropores or nanopores within the soil particles or rock matrices are less bioavailable, and non-aqueous phase liquid fractions that remain soft to mineral surfaces are also less bioavailable, and limited diffusion from solid to aqueous phase also affects the bioavailability. And next factor that controls bioavailability is the mass transfer limitation. So mass transfer has lot of factors involved in it, like the diffusion rate from contaminated matters, how well the pollutant can get desorbed from the soil or sediment particles, and how far it can uh, dissolve in the aqueous matrix in which the microorganism is present. All these mass transfer um, criteria controls the bioavailability of pollutant to the microorganism. And then we have the chemical structure factors where molecular accessibility is very important, and which means that the uh, key bonds that are to be broken in the pollutant should be accessible to the enzymatic or microbial attack. And then steric hindrance plays a important role where substituent groups like chlorine can block the enzymatic sites and hence prevent the enzymatic action. And other chemical structure factors include membrane permeability, where the compounds must pass through the cellular membranes. If not, it's not going to be bioavailable. So physical limitations, mass transfer limitations, and chemical structural limitations are all happening and controlling the bioavailability. So, how does bioavailability affect the degradation of different compounds? For example, when we talk about BTX compounds, which is the benzene, toluene, ethylbenzene, and xylene compounds, they are highly bioavailable, and these compounds are relatively water soluble, and bioavailability rarely limits their degradation. Whereas when you talk about this PAH, polyaromatic hydrocarbons, and polychlorinated biphenyls, they pose significant challenges for microbial access because they have very low bioavailability. So the strategies that we can use to enhance bioavailability is surfactant production, direct contact mechanism, and enzyme secretion, like extracellular enzyme secretion. All these are the three strategies which the microorganisms use to increase the bioavailability. So the next topic is about the enzyme induction and regulation. Enzyme induction represents a regulatory mechanism in biodegradation on how effectively the microorganisms can express the specific enzyme for degradation. Like as we all know, microbes have a very high number of genes in their DNA, and not every gene has to be expressed. So when the microbe is living in a particular contaminated site, it should have the capability to synthesize the enzyme that is going to degrade the contaminant or pollutant in which in the habitat where the microbe is present. So that specific expression is called induction. So based on this, you have different types of enzymes in biodegradation. One is the constitutive enzymes. The constitutive enzymes work in the central carbon metabolism phase B, and they are always present in the microbial cells, like housekeeping enzymes, which are always present in the cell. This includes enzymes involved in the tricarboxylic acid cycle and glycolysis pathway. They generally support uh, general metabolic pathway. So constitutive enzymes are going to be more associated with your phase B biodegradation. When you talk about these inducible enzymes, they are specific degradation enzymes and are produced only when there is a requirement for the action of that enzyme. And these mostly make up the enzyme pool of phase A biodegradation reactions. And in order for them to be expressed and synthesized, we need to have specific induction signals. So you can have different induction mechanism. One is the positive regulation, or uh, the action of inducers. So you can have something like a substrate induction, where uh, when the organism can sense that a particular pollutant is present, it can automatically get the signals for the corresponding degrading enzyme to be synthesized. So this is substrate induction, where the substrate acts as the inducer for the synthesis of the corresponding degradative enzyme. So in case of low concentrations of organic pollutants, the pollutant itself can act as the inducer. And the inducer is going to mainly focus on activating the transcription of uh, genes which code for the degradative enzyme. In case of negative regulation, you call the factor responsible as a repressor, like for example, catabolite repression, where the presence of easily metabolizable substrates is there. Let's say we have a pollutant along with glucose. So it means that the organism is going to prefer eating glucose over that organic pollutant, and your pollutant is not degraded at the end of the day. So this is called catabolite repression. So you can see an example here where ethanol presence can repress the expression of aromatic compounds and degradation genes. Uh, Here the intrinsic repressor molecules block the gene transcription because there is a competition for substrate utilization, and the microorganisms prefers to eat the simplest available substrate than going over breaking down a complex organic pollutant. So this is about positive and negative regulation. Positive regulators are known as inducers and the negative regulators are known as suppressors. So the factors that can affect the enzyme functionality include environmental conditions, like pH sensitivity, temperature effects. So when you go about plotting temperature and pH effects for enzyme, you will get a bell-shaped curve, and the peak of the bell is going to be the optimal condition. It is going to correspond to the optimal pH or temperature value. Below that condition, you are going to have a lesser efficiency, and after that condition also you are going to have lesser efficiency. And you have other inhibitors like heavy metals which can inactivate the enzymes, or again you can have organic compound interference, where the organic compound itself is going to act as the inhibitor for the enzyme function. Similarly, you have other cellular factors where the cell should be in perfect shape for it to carry out its functions. So the proper protein folding and post-translational modifications are essential. You need to have cofactors based on which the enzymes show their performance. The cofactors are very essential for the enzymes to act properly, and you have to have enzyme stability to protect them from degradation. Like when the environmental condition is changing, there is chances of this enzymatic degradation. So the enzyme should be stable even though there is some fluctuations in the environmental condition. This phenomenon is known as enzyme stability, and you have to have clear idea about the cellular energy status. So when there is how much energy expenditure is there, how it is being sorted out by the microorganism, all this you have to know, because this affects the enzyme synthesis and maintenance. The first priority for the microorganism is to go about keeping its routine, or the housekeeping done, and then only it comes to pollutant or contaminant degradation. So hence we need to have a clear idea about the energy status. And then comes the regulated networks, where the expression of degradative enzymes often involves regulatory gene networks controlling multiple enzyme systems. So here you have a cascade regulation where the early enzymes produce inducers for later steps. Like we saw for an enzyme, the substrate itself can act as an inducer. When we are going about a multi-step process, for let's say the enzyme responsible for the third step to get expressed or induced, you need the product of the second step being present in the system. Only then the third step's enzyme will get induced, or the production of the enzyme responsible for the third step will be induced. So hence this acts like a cascade regulated process. And when we talk about inducers, we also have to talk about this repressors or inhibitors, where when you have more of one substrate, it's going to act as a feedback inhibitor, preventing the production of overproduction of enzymes in the previous step. So such kind of regulator network is very essential to ensure an optimal enzymatic process. So the practical implications of this concept is in bioremediation optimization, where we ensure that we supply proper substrate, proper environmental condition and all, and we ensure that we are going to avoid catabolite repression, and if needed we can devise our bioargumentation strategy based on the metabolic network, and we can perform the process monitoring also. So next we'll discuss about xenobiotics and microbial reduction. I'll again pause for a minute so that if you have any doubts, please post in the chat box. Okay, now we'll start discussing about the xenobiotics and their microbial reduction. So xenobiotic compounds are one of the most challenging aspects of biodegradation because they contain structural features which are foreign to the natural biological system, and hence the microorganism is not much acclimatized in breaking these compounds or breaking the bonds in these compounds. uh, Xenobiotics in general is a foreign compound, so it doesn't mimic the natural biological system, and hence the microorganism is not going to have the capability to directly break down the xenobiotic compounds. So why is it essential for us to recognize xenobiotic compounds? So the greater the difference in the chemical structure between a target pollutant and the constituents of living organisms, less likelihood that the contaminant will be recognized as a degradable substance by microbial system. So this is the fundamental uh, principle behind studying xenobiotic compounds in our biodegradation system. So when our target pollutant and the xenobiotic compound are going to be structurally very different, then the microorganism does not identify the xenobiotic compound as something that can be biodegraded. It will only focus on the pollutant. But this xenobiotic compound can still act as a toxic toxicant to the microorganism. And these xenobiotics can be natural or synthetic compounds. In natural hydrocarbons are readily recognized because microorganisms have co-evolved with uh, petroleum derived compounds over geological timescale. In synthetic modifications, we have synthetic modifications of natural structures and which can render the compounds unrecognizable to microbial enzyme systems. Novel chemical architectures present challenges for cellular recognition and metabolic integration. So when we talk about this enzyme substrate relationship, it's more kind of a lock and key mechanism. So if for an enzyme to recognize and degrade a particular pollutant, it needs to have some structural compatibility. If that compatibility is not going to be there, then the enzyme cannot recognize the compound as something that can be acted upon. So you will not get the degradation of these compounds, and all these compounds are said to be foreign to the natural habitat, and hence you call them as xenobiotics. And next comes the concept of xenophores. So xenophores are structural features in organic compounds that are uncommon in nature and make compounds recalcitrant to microbial degradation. For example, we have halogen substitutions like chlorine, fluorine. Chlorine is generally present in pesticides and industrial solvents, while fluorine is extremely stable because it's inert and extremely stable and difficult to clean. When you have substitution in which you have multiple halogens, it's going to increase the recalcitrance further. Other xenophobic groups include nitro groups, cyano groups, and sulfonate groups. So the nitro groups is present in explosives and industrial chemicals, whereas the cyano groups in synthetic compounds, and you can find this sulfonate groups in detergents and dyes. So how these compounds exit recalcitrance? One is by uh, inducing enzymatic recognition failure, where the active site is incompatible and hence the xenophores don't fit the system. As I told, this is going to be a lock and key mechanism, and when there is no proper fit between the lock and the key, you cannot open the door. So in the same way, you have a incompatibility in the active site so that the substrate cannot go and bind to the enzyme, and hence there is no degradation taking place. So there is going to be substrate binding failure and catalytic mechanism disruption. And then you have metabolic integration problems where you have unknown metabolic pathways for processing xenophoric products, which can present us with toxic intermediates that further inhibits the metabolic process. Because of this, the energy barrier increases, and hence the entire degradation process becomes thermodynamically unfavorable. So in metabolic integration, what exactly happens is you have a unknown metabolic pathway which yields some toxic intermediate, and this toxic intermediate is going to highly inhibit your microbial activity, and because of uh, this toxic nature, the energy barrier is going to increase to extend that the degradation process becomes thermodynamically unfavorable. So until and unless you play some with some or the other factor to bring back the feasibility of the process, you cannot expect any biodegradation in a given condition. The factors that influence this recalcitrance of xenophore include the structural complexity, environmental factors, and evolutionary pressure. When you talk about structural complexity, we have multiple xenophores that can increase the difficulty in degradation. We can have substitution patterns that affect the enzyme accessibility. So basically what we saw in the previous slides. You can have multiple xenophores, you can have very rigid substitution pattern, and you can have compounds with very high molecular size and steric hindrance, which is basically the structure based hindrance for enzyme substrate binding. And then you have environmental factors like concentration effects. Either the xenophore concentration is too high or too low, either of it can affect the process. You have co-contaminant interactions where the toxic effects of mixtures is producing a inhibition to the degradation process. And then finally like conditions like temperature, pressure, etc. affecting the microbial activity. You also have this evolutionary pressure that influences recalcitrance. When the exposure time is longer, the microbe is able to adapt better. This is because the selection pressure intensifies and leads to abundance of that uh, particular pollutant. And there is also possibility of genetic exchange between microbial populations such that a positive genetic exchange can support the growth of both organisms involved in the exchange. So such kind of microbial interactions are also possible, but to study these we need to have a little larger time frame than conventional, because we are going to study the effects of uh, evolutionary pressure. So next we'll talk about syntrophic metabolism. I think in one of the previous slides we have already seen about syntrophic metabolism. It is a cooperative strategy where we have different microorganisms or different microbial species, and all of this work together. Primarily there happens a metabolite exchange between these microbial species, and the end result is going to be complete degradation. When you have specific individual microorganisms, you will not achieve the degradation, but you need all this different microorganisms to act in a cooperative manner in order to bring the complete degradation of the pollutant. Here, one organism produces metabolites that serve as the substrate for another organism, and by this, the organisms try to make the thermodynamically unfavorable reactions into feasible reactions. So, as a result of which, we get enhanced degradation, and the key factor here is the interspecies metabolite transfer. So, we have an organic pollutant. Let's say in the first step, we have an organism A that converts the organic pollutant into one intermediate. So there is another organism B that can use that produced intermediate as its substrate, but not the original pollutant itself. So in that case, you can imagine the multi-step enzymatic process kind of a thing only, but now it is a multi-step microbial process itself, where different microbes are required for the complete degradation of the organic compounds. So the thermodynamic basis for centrophic metabolism explained is some biodegradation reactions are thermodynamically unfavorable when performed alone, and the product accumulation can further inhibit the degradation. So here the electron acceptor limitations restrict the complete oxidation. The pH and redox changes from metabolic products can inhibit single organism degradation. And next you have these advantages. So this is the limitations of individual process. One is that the process is thermodynamically unfavorable. Second one is there is going to be a accumulation of a certain kind of product, and third is going to be electron acceptor limitations, and fourth is going to be pH and redox changes because of the accumulation of a single kind of product. So how does syntrophy help us in such situations? It facilitates the removal of the products by partner organisms and further driving back the reaction to be thermodynamically favorable, and it uh, offers maintenance of optimal conditions through metabolite consumption. It facilitates electron flow through hydrogen transfer, and it results in enhanced energy conservation through cooperative metabolism. So what are the key metabolites that are present in such syntropic systems? So this includes hydrogen, acetate, and organic acids. In case of hydrogen, it's a universal electron carrier between the species where rapid transfer due to high diffusion rate occurs, and there's going to be the thermodynamic driver. Uh, it's going to be a thermodynamic driver because low partial pressure needs to be maintained by the organisms that consumes this hydrogen, and there's going to be redox coupling between different metabolic processes. And then you have acetate, which is a major organic intermediate in anaerobic systems. It can act as a carbon and energy source for acetotrophic methanogens, and it's a product of various fermentation pathways. It acts as a central hub metabolite and connects different degradation pathways. And next you have different organic acids like propionate, butyrate, lactate, formate, etc. And you have succinate, malate, all of it, which can enter the TCA cycle. So the factors that affect the syntrophic systems include environmental conditions, microbial community factors. So in case of environmental condition, it's going to be temperature. Temperature mainly affects the metabolic rate and species interactions. And you have pH, which influences the metabolite speciation and microbial activity. And then you have redox potential, which determines the availability of electron acceptors. And then you have nutrient availability, nitrogen, phosphorus, trace elements, and then you have toxic compounds which can disrupt sensitive syntrophic uh, relationships. And then you have microbial community factors like species composition. What are the different microorganisms that are present and what is their metabolic capability? And then you have spatial organization where the proximity affects the metabolite transfer efficiency. So when the organisms are near, then process can be faster. When they are far away, still there is going to be some thermodynamic or chemical limitations for this entropy to happen. That is what spatial organization deals about. And then you have population ratios, where you have a balance between the producers and consumers. So let's say your main pollutant is degraded into a product A, then A is converted to B. So the organism that converts the pollutant to A and the organism that converts A to B should be in a proper ratio. If that's going to be imbalanced, then either the production of A is going to be higher or the production of B is going to be a little bit problematic because A is lower than B. It can happen either way. So hence you have to maintain a proper population ratio, and then you have adaptation time where you have this establishment of stable syntropic relationships. You need to have some time for the microorganisms to establish a proper syntropic relationship. So with this, we will go to specific degradation pathways. I'll just wait for a couple of minutes for you to see if there are any doubts. So next we'll see about the aerobic biodegradation pathway. So in the aerobic biodegradation pathway, it is the most efficient biodegradation pathway for complete mineralization of organic pollutants. So here you have both the utilization of oxygen for metabolic processes and as a terminal electron acceptor. So the aerobic microorganisms degrading the organic pollutants must optimize contact between microbial cells and hydrophobic organic pollutants. Since hydrocarbons are water insoluble, the organisms produce surfactants to make these hydrophobic pollutants accessible to these microorganisms. So here one type is the aliphatic hydrocarbon degradation. Under aerobic conditions, aliphatic hydrocarbons like alkanes, alkenes, alkynes undergo oxygenation to form alcohols. Alcohol dehydrogenases then convert these alcohols to aldehydes, which are then again further oxidized to fatty acids. These fatty acids then undergo beta oxidation, which is the phase B or the central metabolic process that produces the acetyl CoA molecules, and then you reach the TCA cycle. So here you have four important steps. So in case of aliphatic hydrocarbon degradation, when you do this degradation in the presence of oxygen, you have alkanes, alkenes, and alkynes, and they undergo oxygenation to form alcohols. So once you have alcohols, you should have dehydrogenase enzyme to remove the hydrogens and convert this alcohol to aldehydes. And then there should be a step where this aldehyde is oxidized to fatty acids. So these fatty acids can undergo something called beta oxidation and produce acetyl CoA, which is then taking part in the tricarboxylic acid cycle. And this process involves four main steps. One is the initial intracellular attack through oxidative reaction. Second one is the activation and incorporation of oxygen within hydrocarbon molecules. Third is the peripheral degradation pathway conversion to central metabolic intermediates. And finally, you have the biosynthesis of cell biomass from central precursor metabolites. This is about aliphatic hydrocarbon degradation, and next you have this aromatic hydrocarbon degradation. In the aerobic degradation of aromatic compounds, the key intermediate is catechol. So when we saw about this phase A, phase B, we saw there is the occurrence of a funneling mechanism where you have a complex organic pollutant that is being converted into a compound which can enter the common metabolic pathway in phase B. So hence this acts like a funneling mechanism. They are converted into single intermediate. So in this, the initial step involves the hydroxylation of this aromatic compounds like benzene catalyzed by dioxygenase incorporating 2 moles of oxygen. So dioxygenase, you can assume that it's going to incorporate 2 molecules of oxygen to form diol. And this diol component is then converted to catechol by dehydrogenase enzyme. So this is a multi-step enzymatic process. And then the catechol ring is going to be cleaved. And this cleavage can be of two types. It's going to be oxidative cleavage. It can either be a ortho-oxidative cleavage or a meta-oxidative cleavage. In ortho-oxidative cleavage, you have this catechol being converted to succinate and acetyl CoA, whereas in the meta-oxidative cleavage, you can find this catechol being converted to acetaldehyde and pyruvate. All these, whatever may be the cleavage, all these products are the important intermediates in carbon metabolism, and they can directly enter the metabolic path. So this is what is the funneling mechanism, where monoaromatic and polyaromatic and substituted aromatic hydrocarbons are all oxidized and converted to catechol. And this catechol is then converted to acetyl-CoA, succinate, pyruvate or acetaldehyde, which can enter the TCA cycle or other central carbon metabolic cycles. And then you have this role of oxygenases. So the role of oxygenases is very important because we'll see about different types of enzymes in the further slides. But as far as this catechol degradation is considered, oxygenases are very critical because they activate the hydrocarbons for further metabolism, like dioxygenases, monooxygenases. They have remarkable diversity. They So basically the role of oxygenase is to facilitate the addition of oxygen to the hydrocarbon molecule, so that it is very easy for the further metabolic process or degradation to take place. So now we have seen about this aerobic biodegradation pathway. Now we will see about this anaerobic biodegradation pathway. So in the anaerobic biodegradation process, the fundamental difference is that we don't have oxygen as a metabolic tool as well as a terminal electron acceptor. So here we are going to have some alternative electron acceptors and different enzymatic reactions. So the anaerobic process generally proceeds more slowly and it may not achieve complete mineralization as efficiently as aerobic process. In case of aliphatic compound degradation through anaerobic pathways, the aliphatic compounds undergo hydration and carboxylation instead of oxygenation. Because there is no oxygen involved here, they undergo hydration and carboxylation. This leads to the formation of alcohols, fatty acids, and then which then undergo similar reactions in anaerobic conditions, producing oxidized products including carbon dioxide and methane as the final products. In case of aromatic compounds, uh, they show remarkable diversity compared to the uniform catechol pathway in aerobic conditions. So we'll see just two such pathways. One is the benzyl CoA pathway, where the hydrocarbon molecule is degraded to intermediate called benzoyl CoA, which serves as the central intermediate for further degradation. And another pathway through which anaerobic biodegradation of aromatic compound happens is a ring reduction pathway, where the aromatic breakdown occurs through ring reduction mechanism. Like we have different acids as the products produced through ring reduction pathway. These acids further undergo fission and mineralization to carbon dioxide. So we have diversity in the intermediates produced through anaerobic degradation pathway. This is very much contrasting to the funneling mechanism that we have studied in the aerobic degradation pathway. And also this anaerobic pathway is metabolically very flexible to utilize different electron acceptors like nitrate, sulfate, iron or carbon dioxide. uh, but though the flexibility looks very promising for biodegradation, this also implies the complexity in the metabolic and enzymatic um, reactions that are involved. So the environmental implications of anaerobic processes is that the diversity makes it more adaptable but also more complex to predict and optimize. So next we will see about growth associated and co-metabolism. In growth associated biodegradation, the organic pollutants can serve as the sole source of carbon and energy, resulting in complete mineralization. Microorganisms use hydrocarbons as their primary food source, carbon source and energy source. These are predominantly heterotrophic bacteria that have evolved the ability to metabolize hydrocarbon compounds. So the growth associated process involves several key features, like organic pollutants provide the precursor molecules ATP, energy and electron. The organic pollutant itself is going to act as the molecule that is going to give us the precursors, give us the energy, give us ATP and electrons. Microorganisms grow profusely using these resources from pollutant degradation. And complete mineralization occurs because we can break down the pollutants to carbon dioxide, methane, and water. The degradation also directly supports the cellular biosynthesis and reproduction. Microorganisms transform alkanes, aliphatic, and aromatic molecules into substrates that can be funneled into central metabolism. For example, alkanes are converted to fatty acids through beta oxidation, producing acetyl CoA molecules that are oxidized through Krebs cycle. So, whenever the pollutant is going to support the growth of the microorganism, like by acting as a carbon, energy, and electron source, then we call it as a growth associated metabolism. Whereas when we talk about this co-metabolism, it is the kind of metabolism in which the presence of another growth substrate that acts as the carbon and energy source is very essential. So there are going to be two substances. One is the pollutant, and another one is the um, carbon and energy source. So the organism is going to derive energy from one compound, and it is also going to degrade another pollutant that is present in the system. So here the pollutant itself is not the target substrate, but it is degraded due to the lack of enzyme specificity. The mechanistic differences is in growth associated degradation, we have dedicated metabolic pathways where the pollutant serves essential nutritional and energetic functions, whereas in co-metabolism, it relies on only the enzyme activity where the pollutants are transformed by enzymes evolved for different primary substrates. So practically, growth-associated biodegradation results in complete and efficient contaminant removal because the microorganism is going to directly be benefited by the degradation process. Whereas in the case of co-metabolism, it is a very very slow process, and it may be incomplete because it's not the main job of the microorganism to go for degrading the contaminant or the pollutant. So as discussed, we will see about the different enzyme classes in biotransformation. So enzyme classification, we have this EC number. So first type is the oxidoreductases, which we are very much familiar with. We know many all metabolic reactions are going to be either oxidation or reduction reactions. Oxidoreductases represent the largest class of enzymes catalyzing the oxidation reduction reactions, and they are responsible for the most common reactions in biodegradation. This prominence reflects the fact that organic pollutant biodegradation is primarily an oxidation process. Common oxidoreductase enzymes include dehydrogenases, which are capable of removing hydrogen atoms from organic molecules, and oxygenases, which are going to activate the hydrocarbons to facilitate their further degradation. And the oxidoreductases also show different, remarkable diversity where they can act upon different hydrocarbons. So the second type talks about the hydrolases. So hydrolases constitute the second largest class of enzymes and performs hydrolysis of CO, CN, and some CC bonds. They are found in amides, esters, phosphate esters, monohaloalkanes, and epoxides. Important hydrolase examples include esterases, which can break down ester bonds, polymerases that can depolymerize the polymers, dehalogenases that can remove the halogen atoms such as chlorine and replace them with hydroxyl groups. So we have seen that the presence of halogen substitution and steric hindrance are all going to affect the biodegradation process. So when this hydrolase is able to remove the halogen and replace it with hydroxyl group, biodegradation uh, process can happen through normal oxidation mechanisms and get affiliated to the central carbon metabolism. Third type of enzymes include the lyases, which perform non-hydrolytic removal of functional groups from substrates. So it is not going to hydrolyze the functional group, but it's just going to cut the functional group off and remove it from the substrate, resulting in transient formation of double bonds or catalyzing reverse reactions. So, while talking about lyases, decarboxylation and hydration are the key steps in biodegradation process that involve the action of lyase enzyme. And then you have transperases. So, transperases, as the name suggests, they are going to transfer functional groups in between organic compounds. And this is focused for cellular synthesis, and some also benefit the detoxification process. Transferases can sometimes be detrimental because like methylation of mercury, arsenic and strontium increases their toxicity. So when the methyl group is going to be transferred from one organic compound to heavy metal like mercury, toxicity is not going to decrease, but instead we are going to increase the toxicity in that case. The next class of enzymes that we will be looking into is the isomerases, which can catalyze the intramolecular rearrangement of organic compound. Uh, so, in one isomeric state, the organic pollutant can be easily biodegradable, whereas it is very slow in other isomeric state. So, in that case, isomerase enzymes can clearly catalyze the rearrangement of the isomers, transforming them into structures that are more amenable to oxidation. For example, we have enzyme called racemase that catalyze the conversion between L and D amino acids. And then we have ligase enzymes. So the ligase catalyze covalent bond formation similar to isomerases but facilitate subsequent metabolism. So the coordinated function is very important. These enzyme class work synergistically to transform complex organic pollutants. Just when you take a look at the glycolysis pathway, you can understand each type of this enzyme being used in the glycolysis pathway itself. So when you talk about a multi-step enzymatic process, each of the enzyme can be of each of the type that we have discussed, and hence a coordinated function between these enzymes in a synergistic manner is going to drive the entire metabolic process. So next we'll talk about biotransformation, the reaction types in biotransformation. Before that, if you have any doubts, I'll just wait for over a minute for you to post it in the chat box. Okay. So the first kind of biotransformation process is oxidative transformation, where the addition of oxygen or removal of electron from organic molecule or the pollutant molecule occurs. And these reactions are fundamental to most biodegradation processes and include several specific mechanisms. For example, hydroxylation of alkanes, where addition of hydroxyl groups to aliphatic hydrocarbons convert them to alcohols, which can further be metabolized. Second one is the alkylation of compounds, where the modification of aromatic compounds like hydrazine happens. Third is the epoxidation, where the formation of epoxide intermediates from compounds like trichloroethylene happens. Finally, you have this oxidative ring cleavage, where the breakdown of aromatic ring structures and compounds like benzene and other aromatic hydrocarbons occurs. These oxidative transformations typically represents the initial attack on the pollutant molecules, making them more susceptible to further degradation. And the second one is the reductive transformation. So in the case of reductive transformation, it involves the addition of electrons or removal of oxygen from the organic molecules, and they are particularly important for certain classes of pollutants like reductive dehalogenation, nitrile group reduction. Uh, so, in reductive dehalogenation, removal of halogen atoms from halogenated compounds, uh, removal of halogen atoms from halogenated compounds, replacing them with hydrogen atoms, and in nitrile group reduction, reduction of. So in nitrile group reduction, you have the reduction of nitrile groups in compounds like nitroaromatic substances, uh, uh, RDX and TNT. So here the reductive transformation is significant under anaerobic conditions where the electron acceptors other than oxygen are utilized. And then you have hydrolytic and other non-redox biotransformation, and these transformations do not involve electron transfer, but they involve bond breaking and formation through other mechanisms. For example, hydrolysis. Hydrolysis is water-mediated cleavage of bonds in esters, amides, and other compounds. Next is the hydroclimatic reactions, where addition of water molecules across double bonds happens. And next is the dehydrogenation reaction, where you remove hydrogen without electron transfer. And next is the hydrohalogenation, where there is removal of hydrogen and halogen atoms simultaneously. They often prepare the molecules for subsequent oxidative or reductive transformation. And next you have synthetic reactions, which involves the formation of synthetic reactions in the sense there is some synthesis happening, which is like formation of new bonds and incorporation of pollutant derived molecules into cellular components. For example, carboxylation, addition of carboxyl group, methylation, addition of methyl groups. So you're building something new. These reactions enable the incorporation of pollutant derived carbon into anabolic reactions and biomass synthesis. So, you are taking the carbon from the pollutant and integrating it into the microbial system. So, you are catabolizing it first and then going on to anabolic reactions, integrating that into anabolic reactions. The last one is this integrated function, where all these four types, which is the oxidative, reductive, hydrolytic, and other non-redox and synthetic, all together they work in combination to achieve complete pollutant mineralization. The initial oxidative or reductive attack prepares the molecule for further transformation. Hydrolytic reactions modify functional groups, and synthetic reactions incorporate useful fragments into cellular metabolism. So we have seen about this aromatic compound degradation and catechol pathway earlier. So I'll just directly skip to the microbial adaptation and pollutant bioavailability topic. So microorganisms have evolved sophisticated strategies to adapt organic pollutant environments and overcome the inherent challenges of pollutant bioavailability, particularly for hydrophobic compounds, because hydrophobic compounds like the hydrocarbons from petroleum industries are very much less bioavailable. So for this, they use different adaptation techniques. One is the evolutionary adaptation. Here, although the hydrocarbons like polyaromatics monoaromatics and alkanes are not regular substrate for many microorganisms. The microorganisms can accustom itself in utilizing these, and it has the evolutionary capacity. So this stems from the natural occurrence of similar molecules during evolutionary process, providing the foundation for uh, enzymatic recognition and transformation. The microorganisms gradually transform alkanes, aliphatic and aromatic molecules into potential substrates. This can be funneled into the central metabolism, and this provides a greater metabolic flexibility that allow bacteria to uh, go about utilizing many different novel uh, carbon sources. And next you have this bioavailability challenges. So here organic pollutants, particularly hydrocarbons, are often hydrophobic and water insoluble. So hence their bioavailability is very low, and the access of the pollutant to the microorganism is uh, the limiting factor. So here, the compounds tend to partition into non-aqueous phase associated with soil particles or form separate phase liquids, making them inaccessible to aqueous phase microorganisms. The hydrophobic nature of pollutants create a fundamental challenge where the microorganism must establish contact between their aqueous cellular environment and the hydrophobic pollutant molecules for biodegradation to occur. And next you have a mechanism where the organism goes for biosurfactant protection. So these biosurfactants are amphiphilic molecules that can reduce the surface tension between the hydrophobic and aqueous phase. So how exactly the biosurfactant works is first, it brings about emulsification. Biosurfactants create stable emulsions of hydrophobic pollutants in aqueous media. They increase the solubility of the hydrophobic compound by and then facilitate the transport of pollutants from non-aqueous phase to microbial cell surfaces. Some biosurfactants modify cell surface hydrophobicity to enhance pollutant adhesion. So this is about the evolutionary adaptation. So in metabolic adaptation, first is enzyme induction. So microorganisms can induce specific enzyme systems in response to the pollutant that is present in the system. So based on the pollutant that is present, you can either see a upregulation of oxygenases, dehydrogenases, and other enzymes that is required to degrade the particular pollutant. And next you have this metabolic pathway flexibility, where you have the ability to switch between different metabolic pathways depending on available substrates. Uh, this is one of the crucial adaptations where the microorganisms can utilize pollutants through both growth associated and co-metabolic mechanisms. The next one is in the metabolic adaptation is the broad enzyme specificity. Many biodegradative enzymes exhibit broad substrate specificity, which means they can recognize and transform structurally diverse pollutants. For example, the oxygenases that attack aromatic hydrocarbons show remarkable diversity, allowing conversion of various aromatic compounds to catechol. So next we'll talk about the cellular contact optimization. So till now we have seen about the metabolic adaptations. Now we'll see about the cellular contact optimization, where the microorganisms have developed metabolic processes for optimizing the contact between the cell and the pollutant. For example, chemotaxis, which is the movement. So based on the signal from the pollutant, there will be a directional movement of the microorganism. Next one is the adhesion mechanism, where the pollutant and attaches to the surface of the microorganism. And third one is the biofilm formation, where there is a formation of a biofilm where you can see a high concentration of enzymes that is capable of degrading the pollutant. You also have another strategy called the co-metabolic strategy, where the evolution of co-metabolic capabilities represents another adaptation strategies. So enzymes like methane monooxygenase uh, evolved for specific substrates can transform other compounds like TC due to their broad specificity. Okay, so we have seen about evolutionary adaptations, uh, and then about metabolic adaptations. So now we will see about physiological adaptations, where microorganisms may alter their cellular composition and physiology to better tolerate pollutant exposure, including modifications to membrane composition, stress response systems, and detoxification mechanism. And you, which is the physiologically how microorganisms can adapt. Last one is the community level adaptation, wherein the microbial communities are going to work synergistically. So the entire community should get adapt to degrade a particular pollutant or mineralize a particular pollutant. So this is the community level adaptation. So in the last part of today's thing, we will see about the um, metal biotransformation. So first, for that, we need to know about the categories of metals based on their physiological significance. Um, which includes essential non-toxic metal, essential harmful at higher concentration metal, and always toxic metals. So the first category, which is the essential but basically non-toxic metal, includes calcium and magnesium. These metals are required by the organism for its normal biological functioning, and while it is not toxic to the microorganism even when it is present at a higher concentration. The category 2 is the essential but harmful at higher concentration. So this includes iron, manganese, zinc, copper, cobalt, nickel and molybdenum. These metals are required in trace amounts for the microbial growth, whereas when surpass a certain optimal concentration, they are going to be detrimental for the microbial growth. That is category 2. And finally, you have always toxic metals, which includes mercury and cadmium. No matter how much concentration they are going to be toxic to the microorganisms because they are not involved in any biological function as such, and they are not essential for the growth of microorganism. So the cell can respond in different ways to these toxic metals. So the cell has no desire to uptake these metals because these metals do not serve any biological purpose. However, uh, because of the metallic nature, just through the conventional routes, these metals can also go into the microbial cells. So once this is sensed, this microbial cells immediately go upon activating the defense mechanism, which includes turning on the efflux systems, going on to biotransformation processes, or activating some sequestration mechanism. So here the cellular response is always defensive, unlike essential metals where initial response may be uptake oriented. So next we will see about the mechanism of metal microbe interactions in bioremediation. First one is the bioaccumulation. In this, there is active transport of divalent metal ions inside the cell using general or specific transporters. Large number of metal ions are deposited or accumulated within the cell. This is an energy dependent process requiring ATP or transmembrane gradients, and it is a property of living cells. The effectiveness is it is highly effective for concentrating metals from dilute solutions. Applications include it is suitable for waste volume reduction, particularly in case of radioactive metals, where storage is becoming a major concern. So the next mechanism is biosorption, where it is a metabolism independent physicochemical process where these cations are physically or chemically adsorbed onto the cell surface. This involves the interaction using different functional groups. It includes both surface absorption and gradual absorption into the cell envelope. So it can be performed using dead or metabolically inactivated cells also, and this can make this process cost effective. And it is useful for treating contaminated water systems, and it does not require maintaining viable cell population. So next is the biotransformation process. This is a redox driven process, and it converts the oxidized or reduced metal into different forms. For example, you have uranium 6 converted to uranium 4, chromium 6 converted to chromium 3, arsenic 5 converted to arsenic 3. All the 6 4 is all the oxidation state. From oxidation state of 6 to 4, if uranium is converted, it's a kind of a biotransformation. So basically, when the oxidation state changes, everything including the solubility, toxicity, mobility, all this changes, and hence the amount of damage that we see in the environment is also going to change. So that is the biotransformation process. It is highly effective for immobilizing the metals through precipitation, and applications include chromium and uranium remediation in groundwater, where reduced forms precipitate and immobilize contaminants. And the next mechanism that you can work on is the extracellular complexation or precipitation. This involves cellular ligands or molecules like polysaccharides, metallothioneins, siderophore like compounds, etc., that are produced and released by the microorganism. These molecules complex with the metal outside the cell, leading to precipitation. It creates a stable metal complex that are less bioavailable, and it is useful for in situ remediation where metals can be immobilized in contaminated sediments or soils. So the goals that are achieved through microbial bioremediation of heavy metals include precipitation and immobilization, which we have discussed in the last part in the previous slide, volume reduction of contaminated matrices, because when you're going to precipitate some metal, you can do the solid liquid separation and everything, and finally you will end up with volume reduction of the contaminated matrices. Compartmentalization of harm reduction. So this involves using microbial processes to transform metals and to compartmentalize them in environmental locations where their mobility is arrested, and hence the harmfulness is also reduced. So this is compartmentalization for harm reduction. So in contrast with organic pollutant remediation, we'll just see a comparison between organic and inorganic pollutant remediation. So organic pollutant, we can see complete mineralization, whereas in inorganic, you can only see transformation. Organic pollutant is completely eliminated, whereas inorganic pollutants are only contained and not eliminated completely from the systems. The metabolic energy utilization for organic pollutants is it often provides metabolic energy to microorganisms, whereas in metal bioremediation it requires energy expenditure for the transformation process. Organic pollutant remediation results in elimination of contaminants. So metal remediation requires long-term stability, consideration of long-term stability of immobilized or transformed metal forms, as changing environmental conditions could potentially remobilize the metals back. So we are just changing the state. It can anytime change back to its original state. So with this, we conclude today's session on biodegradation and bioremediation. If you have any doubts, I'll just wait for 2 minutes. You can post in the chat box, or you can feel free to leave the session. Thank you so much for your patient listening. If you have any doubts, post in the chat box. Otherwise, please feel free to leave the session. Thank you so much for joining today's session. We'll end it here now and we'll discuss further in the next session. Thank you so much.